everyone, have a good day. I am Jessica Isimbahon, and today I will going to discuss another topic. This is all about microbial metabolism. Please like and share, and if you are new here, don't forget to click the button and subscribe. Microbial metabolism in extreme environments has two defining characteristics. It is slow and it tends towards the lower measurement threshold of all current techniques and methodological terms. Also, in a typical environments include microscopy at surface air interfaces, mass balances, radio-labeled fatty acid synthesis. Microbial metabolism are described all of the chemical reactions inside the cell is metabolism. The metabolic processes are all of the processes inside the cell or living organism that are necessary for life. Enzyme or proteins are specific to what enzyme they catalyze. For example, glycogen synthesizes glycogen from glucose stored in the liver. The chemical reactants to which an enzyme binds are called substrate lines. Enzyme active site. As an enzyme binds its substrate, the enzyme structure changes slightly to find the best fit between the transition state, structural intermediate between the substrate and product, and the active site, just as a wrapper glucose moves to a hand inserted into it. Factors affecting enzyme activity. Temperature. Reactions occur more rapidly as temperature rises. As long as enzyme is active, heat can denature enzymes. pH. Enzyme structure depends on pH. pH affects charge or R groups, protein structure. Substrate. Reactions occur more rapidly as substrate rises. Saturation occurs when substrate is high enough. Adenosine triphosphate or ATP, preferred source of usable energy for all cells. Breaking band of third phosphate releases ideal amount of energy. Band is easily broken, low energy. This is why organisms convert food energy to ATP energy. How is ATP produced? In most organisms, energy from a food source is converted to energy in ATP by glycolysis, followed by one of two processes: fermentation, low ATP yield, or respiration, high ATP yield. Glycolysis. Glycolysis is a catabolic pathway by which sugars such as glucose and several other food sources are broken down to 2,3 carbon molecules of pyruvic acid or pyruvate. Releases energy to yield 2 ATP per glucose. Also transfers high energy electrons to NAD to yield to NADH. Oxidation reduction. Much of the energy in food molecules such as glucose is captured as high-energy electrons by electron carriers such as NADH and FADH. When a molecule receives or gains electron, it is said to be reduced. Electrons are typically transferred as part of a hydrogen atom. A molecule that gives up electron loses hydrogen is said to be oxidized. Respiration. After glycolysis, energy in pyruvate and NADH is used to produce much more ATP by respiration. Krebs cycle breaks down pyruvate to 3CO2, energy captured as electron by NADH and FADH. Electron transport. Electron from NADH, FADH used to produce hydrogen gradient. Chemiosmosis. Hydrogen gradient used to make ATP. Fermentation. ATP production begins and ends with glycolysis in organisms that ferment. Fermentation is all about recycling NAD+ so that glycolysis can continue. NADH is oxidized to NAD+ by reducing pyruvate to lactic acid, for example. The Krebs cycle. A cyclical metabolic pathway catalyzed by enzymes in the matrix of mitochondria. Requires 2 C-acetyl groups connected to coenzyme A acetyl, 3 C-pyruvate plus coenzyme, 2 C-acetyl coenzyme plus CO2. Krebs cycle. Electron transport and chemiosmosis occurs in the mitochondria of eukaryotes and at the plasma membrane of prokaryotes. Oxygen is usually the final electron acceptor, but other molecules can play this role in anaerobic respiration. All organisms depend on autotrophs. Autotrophs can produce organic molecules from CO2 and inorganic carbon source. All heterotrophs require an organic source of carbon. Organic molecules, directly or indirectly, come from autotrophs. The source of energy for autotrophic processes can be light, photoautotrophs that carry out photosynthesis, chemical, chemoautotrophs that use various molecules as a source of high energy. Light reactions of photosynthesis. Electrons from H2O energized from sunlight fuel the synthesis of ATP through electron transport and chemiosmosis, much like respiration. Ultimately reduce NADP+ to NADPH. ATP and NADPH provide energy to fuel production of sugars in the dark reactions. Dark reactions involves an anabolic pathway known as the Calvin-Benson cycle. Endergonic reactions of this pathway are fueled by ATP and NADPH from the light reactions. Resulting sugars can be used as a source of energy or to build other organic molecules.